certainly on the exam you get a lot of questions on this particular chapter. So I don't know if it's the first exam or it's just a difficulty level of the chapter. The students definitely have a hard time. So you need to spend some time on this chapter. Okay? Not only obviously trying to understand what I'm going to talk about, which is really the skeleton, but also getting the notes in the book and reading the chapter. Okay? It's really important. A lot of stuff is coming in very, very fast. I've split this into two parts. The first part we'll go through metabolism, if you like, more of the exercise biochemistry. And then the second part we're going to look at hormones, endocrinology, so to speak. Okay? If you've had endocrinology classes before, you'll be fine. Okay? Okay, let's go straight in. Metabolism and bioenergetics. Okay? We all know about the various substrates, so if you like substrates, we get some nutrients. Carbohydrates, we're going to talk about today, obviously, fats, and a little bit about protein. Okay? We're going to talk about those chemical pathways that break the substrates down. Okay? And that process is known as bioenergetics. Okay? In fact, there are a few departments around the states, in particular my old department of Colorado State University has a PhD in exercise bioenergetics. So it's a very involved field. Okay, basically it's just all about the chemical pathways that these substrates or breaking the substrates down into usable energy. That's bioenergetics. Okay, and we know that energy that we derive from those substrates is obviously ATP, adenosine triphosphate, or CP. Okay, and we know in most instances that ATP serves as the immediate source of energy, right? We talk about the phosphocreatine system. Okay? And it can also be used to drive other chemical reactions. Okay? And we also know how important it is for muscle contraction, right? Breaking the cross bridge, helping to pump calcium back into the cytoplasmic reticulum, etc. At rest, you sitting there at the moment, right, you're basically using both carbohydrate and fat in equal amounts to take for energy. We know that changes during exercise, we're going to talk about that. Protein provides little energy for cellular activity, okay, unless you're in a starvation state. Okay. When we're talking about intense short duration efforts, okay, probably all know this before, you're relying mostly on carbohydrate, okay, to generate ATP, immediate energy source. Okay. And of course, for more endurance type events, marathons, etc., long training runs, bikes, etc., you're predominantly using fat. Okay. Fat for energy. Okay, your book doesn't talk about this, but it's important. Okay, that's why I put it up there. It's called the crossover concept. What we got here, on the y-axis, we got 3 fatty acids. And glucose, RA, stands for rate of appearance in blood. We can measure that in the lab. Okay, so that's 3 fatty acids, which you get from a triglyceride, or fat molecule. Okay, and glucose, rate of appearance in blood. Down here, we have the percentage of VO2 max. Are we all comfortable with VO2 max? I know you haven't done a lab on VO2 max, and we haven't really spoken about it, but from other classes, are you all happy with VO2 max and what VO2 max is? I don't have to go through it. We will touch on it again, obviously, okay? The maximum amount of oxygen to consume, okay? Okay? Usually measured in litres per minute, or mLs per kg per minute, okay? So obviously, as we're going up from 20, 40 to 60, 80 to 100%, our effort, our muscular error, our intensity effort is increasing, right? So, obviously, at low percentages of VO2 max, okay, what we've got is our glucose rate of appearance, this one right here, okay. We've got 3 fatty acids right here, okay, and we've got the glycogen usage right here. So what's occurring up to about certainly 60% of VO2 max we're predominantly using fat. Right? So our rate of appearance of free fatty acids in the blood is increasing, which you'd expect. Right? After about 60% of VO2 max, okay, what's becoming more predominant? Glycogen breakdown and the rate of glucose appearance in blood. Okay? So this is real data. Okay? So what it's suggesting is that after about 60% of VO2 max, carbohydrates is becoming your predominant source. For that, it's fat. Okay? And that's called the crossover concept because they tend to cross, obviously, at about 60% of VO2 max. Everyone okay with that? Crossover concept. Okay, in terms of carbohydrates then, all dietary carbohydrate is ultimately converted to glucose. We know that the glucose is taken up by muscles and the liver, okay, and converted to glycogen, the storage form of glucose. Where's glycogen stored? It's stored in the cytoplasm of muscle cells, and as we know, it's broken down from ATP. Okay. It is also stored, and we'll see this, it's also stored in the liver, okay, and it's also converted back to glucose. I'll talk about that metabolic pathway coming up. Okay, so it can be stored in liver and it can be broken back down to glucose if you need it. Okay, it's also transported in the blood back to muscle. Okay. Fat, like we said before, provides substantial energy during prolonged low-intensity exercise. The fully stores of fat are larger than carbohydrate, generate more ATP from the breakdown of fat. Fat is stored as triglycerides. Okay. It must be broken down. Typical triglyceride molecule sure it has a glycerol backbone. And fatty acid side chains, right? When this triglyceride molecule is broken down, the fatty acid side chains are cleaved off, right, by lipase, the enzyme. They become your free fatty acids that cleaved off from the glycerol backbone and move into blood. Okay? That's what we mean by free fatty acids. Okay? They can be used for energy, of course, free fatty acids, and the glycerol backbone can be used for energy as well, and be broken down. We'll see that coming up. But it's all usable. Okay? It's all usable. More energy is derived from breaking down fat, 9.4 kilocalories per gram compared to carbohydrate, around about 4.1. This is just a table out of your book here looking at looking at carbohydrates in terms of liver glycogen, muscle glycogen, glucose in body fluids, and fat, subcutaneous and visceral and intramuscular. You can see that we've got a lot more kilocalories coming from fat than we have obviously from carbohydrates. Okay? No great surprise there. Like we said, protein's not used much for energy, certainly during exercise. If it is used, it must be converted first to glucose via a metabolic pathway termed glycogenesis. Proteins can also, believe it or not, generate free fatty acids as well. And that's during a starvation state. Okay, lipogenesis. Supply up to about 5 to 10% of energy during prolonged exercise, but it needs to be broken down first, obviously. And the basic unit, as you all know, is amino acids. Okay, they can be used for energy, but predominantly it's used during a starvation state. If you're extremely low in fat, which you never are, but in terms of the metabolic pathways you can be, and if you're low in carbohydrate, protein can be used. I want to talk a little bit about enzymes and how important they are. Okay? You should know from previous classes, right, enzymes control the rate of free energy release from a substrate. Okay? Enzyme names end with the suffix ase, right, lipase, etc. Okay? That's a giveaway. We're going to talk about glycolytic enzymes and oxidative enzymes coming up. Glycolytic enzymes act in the cytoplasm of the cell. Okay, so there's my cell, right? There's my nucleus. Here's my mitochondria, plenty of them, double membranes. Okay. So when we talk about glycolytic enzymes, we're talking about working in the cytoplasm of the cell, right? When we're talking about oxidative enzymes, they're working in the mitochondria. Okay? Big difference. Okay? And we know, of course, enzymes bind with a substrate. So we talk about this lock and key action of enzymes. Okay? A substrate is broken down. So a substrate is broken down into products, right? Whatever they may be. What controls the rate of that? Enzymes. What's the end result? Free energy. And a lot of other things. Okay? Free energy means ATP. Okay? Some factors that affect enzyme function. Increasing the square brackets means concentration of, right? Increasing the concentration of a particular enzyme increases the rate of reaction. That's what occurs during training. Okay? So training increases concentration of some enzymes. A lot of those glycolytic enzymes have increased with training, particularly with endurance training. Okay? That speeds up the reaction. Increasing temperature in the muscle increases the rate of reaction. If you get too hot, it starts to denature the enzyme. Increasing the substrate concentration also increases the rate of reaction for a given enzyme. If you increase the substrate, that increases the reaction. Okay. So increasing the concentration of the enzyme, increasing temperature, increasing substrate concentration. So you can think about nutrition during endurance, for instance, during endurance events, important, right? You've got to keep the substrate concentration up. Okay? Temperature's going to go up anyway in the muscle. Okay? And by training, you increase the enzyme concentration. Okay. I do want to talk about, very briefly, we could spend a whole lecture on these, allosteric enzymes. These are very, 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 very important. Okay? I'm not sure if your book touches on these or not, they may mention it briefly. What are allosteric enzymes? Allosteric enzymes are very important enzymes, like I said. They are located at or near what we call branch points in metabolic pathways. And they're influential in directing substrates along one or another of the available metabolic paths. Okay, we'll see a little diagram coming up in a minute. Okay. There's also what we call effectors, a whole bunch of effectors that can, if you like, modulate or influence the activity of these allosteric enzymes. So they can activate or inhibit these enzymes. It may be in some instances you want to inhibit a particular metabolic pathway. And an effector will come in and will inhibit the enzyme, the allosteric enzyme. There's times obviously when you want to turn on the metabolic pathway, and there's effectors that will turn on a particular enzyme, allosteric enzyme. An example of an effector, okay, that will turn on an allosteric enzyme is cyclic adenosine monophosphate, or otherwise known as cyclic AMP. We'll see him again under hormones. Okay, so everyone get it? So we've got these allosteric enzymes. There they are again, right? Like I've said, they can be activated or inhibited, i.e. they have increased or decreased activity at a given substrate concentration. So inhibition of a given enzyme without the associated substrate to be directed to another metabolic pathway. So, examples of allosteric enzymes. Phosphorylase is the main enzyme involved in breaking down glycogen back to glucose. So here we've got glycogen being broken down back to glucose 6-phosphate under the action of phosphorylase. This phosphorylase is turned on, switched on, causes glycogen to be broken down. There's many cases where you don't want glycogen to be broken down, and an effector that inhibits phosphorylase will come in and stop phosphorylase from working. Okay. Another example is phosphofructose kinase, otherwise known as PFK. Okay, which is right here. Okay, actually, sorry, I lied. Here, breaking down glucose to glucose 6-phosphate, PFK. And there's others, hexokinase, lactate dehydrogenase. These are all very important enzymes. You turn them on, will break down substrates. Okay, so those are allosteric enzymes. Not every enzymes in these metabolic pathways are allosteric. Okay, that's very important. These ones are very important. Okay, I don't want to get too bogged down with Michaela's mentioned kinetics of enzymes, but I do want to mention that this particular type of kinetics of an enzyme is not relevant to allosteric enzymes. It's relevant to all the other enzymes I spoke about, the intermediate enzymes. So what I've written here is the kinetic model is relevant to situations where the concentration of enzyme is much lower than the concentration of the substrate. In other words, where enzyme concentration is a limiting factor when the enzyme is non-allosteric. Basically what this shows is that if you increase the substrate concentration, which is very easy to do, right? You increase the reaction rate for that particular enzyme. Up to a point, right? Because it starts to bottom and it starts to flatten, right? It starts to flatten. Why does it start to flatten? There's only a certain amount of binding sites to substrate with an enzyme, right? So we've got full saturation here. Okay? Full saturation. So what I've written here is to determine the maximum rate of an enzyme-mediated reaction. A series of experiments is carried out where the substrate concentration is increased until a constant initial rate of product formation is achieved. This is the maximum velocity. Okay, that's the maximum velocity. Right? So adding substrate after that point doesn't change the maximal rate of that concentrate of that enzyme. That's the bottom line. Okay? So we call that a steady state. It's reached a steady state. Okay? So in this state, enzyme active sites are saturated with substrate. It's a very simple experiment to do. We do this in the graduate exercise biochemistry class. Just keep adding substrate and measuring the rate of reaction of a particular enzyme, and you always see this type of curve. Okay? Like I say though, that does not occur with allosteric enzymes, but it does occur with these other intermediate enzymes. Okay? Okay, it's starting to get a little bit noisy. Hopefully it doesn't get any worse than this. There's no key points. I'll let you read the key points like I always do. Now I want to talk about 3 energy systems. I want to talk about the ATP phosphocreatine system, then I want to talk about the glycolytic system, and the enzymes where the enzymes are located in the glycolytic system, in the cytoplasm, right? And I want to talk about the oxidative system. The enzymes in the oxidative system are located in the mitochondria, right? So we're going to talk about those 3 energy systems. Okay? And remember, when you break down ATP, under the action of ATPase, the enzyme, you generate ADP plus inorganic phosphate, PI, plus energy. Okay? So, these cells, right, they store small amounts of ATP and phosphocreatine, which is broken down to regenerate ATP, the phosphocreatine. Okay? Creatine kinase directs this reaction. Does not require oxygen, this reaction, anaerobic. So ATP and phosphocreatine, okay, to sustain the muscle energy needs for only about 3 to 15 seconds. No longer. That's during a nice long out sprint. Okay? Very important, 1 mole of ATP is produced by 1 mole of phosphocreatine. Okay? The energy that's generated from the breakdown of phosphocreatine, right, is used only to regenerate ATP. It's not used for anything else in the cell. It's only used to regenerate ATP. Okay? So what we've got here then is phosphocreatine under the action of creatine kinase, the enzyme. Phosphocreatine is broken down to creatine and inorganic phosphate. That releases energy, and that energy recombines ADP and PI back to ATP. That's it. Okay. So what you see is something like this. Here's the percentage of resting value right here. Here's time. Blue is ATP. Here's your seconds, obviously 14 seconds. Here's your phosphocreatine in red. You can see that initially, obviously, the phosphocreatine is being used up, right? Because it's going down. What's it doing? It's generating ATP, which is doing a fine job up to around about 8 seconds, right? We're still using phosphocreatine up. The ATP stores are starting to drop, and then after 14 seconds we reach exhaustion. We reach exhaustion because we can't break down any more phosphocreatine with only limited stores. That's it. Okay, you can't generate any more ATP. That's the rationale behind creatine loading, right? Phosphocreatine loading, which we'll talk about in ergogenic aids again coming up later on. Okay? You've only got limited amount of stores of phosphocreatine. You've got limited stores of phosphocreatine, you've got limited stores of ATP, you've got limited stores of ATP. Okay. The ATP phosphocreatine system. Now I want to talk about glycogen breakdown and synthesis. Okay? This is where the fun begins. Okay? Terminology that you're going to have to get your head around if you haven't seen this stuff before. It's been a few years. Glycolysis, glycogenesis, glycogenolysis. All very similar sounding, but all very different. All starts with G, so it does get confusing. Okay? So just to start off with in terms of definitions, glycolysis, what is glycolysis? The breakdown of glucose. It may be anaerobic or aerobic. Lysis, as you know, means breakdown. This is how I remember it when I was a student, right? Lysis, breaking down. Not the formation of, right? It's breaking down. So it's the breakdown of glucose. Glycogenesis, genesis, obviously the making of, right? Glycogenesis is the process by which glycogen is synthesized from glucose. Stored in the liver or muscle. Glycogenesis is the process by which glycogen is synthesized from glucose, like glucose. Glycogenolysis, again lysis, the breaking down, process by which glycogen is broken down into glucose 1-hydroxy glucose 6-phosphate used for energy. Okay. So glycolysis, glycogenesis, glycogenolysis. Okay. And there's another one I'll throw at you later on, gluconeogenesis. That's the fourth one. Then you've got lipolysis. Then you've got lipogenesis. There's 6, right? Okay, so let's talk about the glycolytic system, right? The glycolytic system occurring in the cytoplasm of the cell, right? We're not talking at the moment about the mitochondria. Okay. This particular glycolytic system requires 10 to 12 enzymatic reactions to break down glycogen or glucose. When we're talking about glycogen, we're talking about lactic acid, producing ATP. It occurs in the cytoplasm, like I just mentioned. It can be anaerobic or aerobic. If it's without oxygen, pyruvic acid, we're seeing this produced by glycolysis becomes lactic acid. Okay? This is important. We'll go through this. 1 mole of glycogen produces 3 moles of ATP. It's all about ATP, right? 1 mole of glucose produces 2 moles of ATP. And we'll see why that occurs in a second. Okay? And if you combine the ATP phosphorocreatine system and glycolysis, you provide energy for about 2 minutes of all-out activity. That's it. And there's another system that we'll talk about after that takes off, and that's the oxidative system. Oxidative phosphorylation or mitochondrial respiration. All meaning the same thing. Okay. All right. This diagram is not out of your book. It's a similar diagram. The one in your book is small and confusing. I can kind of think it's confusing. It's a similar book this is out of another exercise physiology textbook, so I prefer this one. It's bigger, but it does the same job. Again, this is glycolysis. And we're going to start off with glucose right here. Okay? So we've got glucose, right? Glucose in the cytoplasm of the cell is broken down to glucose 6-phosphate. And it requires energy. ATP is broken down into ADP. So we're down 1 ATP. So you can say minus ATP right there, right? Minus 1 ATP. Glycogen, so also this glucose, when it's broken down into glucose 6-phosphate, is under the action of this under the action of hexokinase. Hexokinase is a allosteric enzyme. It can be inhibited or activated. Okay. Here's glycogen. Glycogen can also be broken down into glucose 6-phosphate. No energy is required. It's under the action of phosphorylase. And if you recall, phosphorylase is also an allosteric enzyme. It can be inhibited or activated. Glucose 6-phosphate is then broken down to fructose 6-phosphate under the action again of this particular enzyme. This is not an allosteric enzyme. This is an example of not an allosteric enzyme. And then fructose 6-phosphate is broken down into fructose 1,6-diphosphate under the action of PFK. That is an allosteric enzyme, and we've used up another ATP. So we're down 2 ATPs. Okay, let's turn the page. We're here, right? Fructose 1,6-diphosphate. When fructose 1,6-diphosphate is broken down by aldolase there, it's broken down into 2 moles of 3-phosphoglycerolaldehyd. 1 here and 1 here. Okay. And this particular substrate is broken down into 1,3-diphosphoglycerate. Okay, we're generating a hydrogen right here, and these are picked up. These are picked up by carriers. These are what we call coenzymes. NAD and FAD. Nicotinamide adenine dinucleotide and flavin adenine dinucleotide. There's two of them. So NAD combines with hydrogen to become NADH, and that goes on to the electron transport chain, which we'll talk about coming up. Okay, and obviously, just as a brief thing, the electron transport chain, that's where the split hydrogen ion is split. Those split into hydrogen ions and electrons, but we'll deal with that coming up. So basically then, from diphosphoglycerate here to phosphoglycerate, we now generate an ATP on both sides, one here and one here. Okay? So we're down -2. We've now reclaimed some ATP, so we're at 0 again. All right? We go on down these reactions, all the way down to here. And again, we're generating another ATP. With the end result that we're making pyruvate. Okay? So an ATP being generated here, an ATP being generated here. Under the action of pyruvate kinase. The reason it's in yellow is because that's an allosteric enzyme. It can be inhibited or activated. If we have oxygen, if this is aerobic, our pyruvate doesn't show it on this diagram, then converts into acetyl-CoA. If it's anaerobic, the pyruvate is converted into lactate under the action of lactate dehydrogenase, which is another allosteric enzyme. Okay? So if you recall, we go back to here, 1 mole of glucose produces 2 moles of ATP, if you recall. Right? We've lost 2 here. We've gained 2 here, so we're back to 0. We gained 2 here, so that's +2. Glycogen, breaking down with glycogen, we bypass this step right here, don't we? That's not involved. So glycogen is broken down to glucose 6-phosphate under the action of phosphorylase. There's no energy required in that. So the net ATP result is 3 ATPs of glycogen. We get more energy to break down the glycogen. Okay? 2. Okay. There's the key points. Okay? Just to go through though, this one, the ATP phosphocreatine system. Energy yield is 1 mole of ATP for 1 mole of phosphocreatine. It's not a lot at all, right? For the glycolytic system, glucose or glycogen is broken down to pyruvic acid. And without oxygen, it's anaerobic, and pyruvic acid is converted to lactic acid. Like we just said, 1 mole of glucose yields 2 moles of ATP, 1 mole of glycogen yields 3 moles of ATP. This is all good for about 2 minutes of exercise. Then you're done. Okay. All right. So like I say, you're good for about, it says about 1 minute. It's about 2 minutes of exercise. And the other system that then kicks in, takes off, is what we call, some books call it mitochondrial respiration, otherwise known as mitochondrial or oxidative phosphorylation. I'm going to call it oxidative phosphorylation. But mitochondrial respiration equals oxidative phosphorylation. So if you're running a marathon, etc., going for a long bike ride, oxidative phosphorylation is your main energy pathway. Okay? And it requires oxygen, of course. Okay, let's talk about the oxidative system now. Obviously, the name suggests it obviously uses oxygen. Okay? In terms of oxidative production of ATP, it occurs in the mitochondria, not the cytoplasm. Okay. can yield much more energy than the anaerobic systems. It is slow to turn on. Okay? And like I've already said, the primary method of energy production during endurance events. But it does work in conjunction with glycolysis. Okay, so for example, here's a very brief overview, very simplified schematic, right? We've got glycogen being broken down to glucose 6-phosphate. That's glycolysis. We've got 3 ATPs being generated, you know that. Okay. The pyruvic acid is broken down to acetyl-CoA and it goes into mitochondria. We're going to talk about the Krebs cycle or the citric acid cycle in a minute. Acetyl-CoA enters the Krebs cycle or the citric acid cycle located in the mitochondria. Remember, we've got 2 moles of acetyl-CoA going into the Krebs cycle. We've got 2 moles of pyruvic acid. That generates 2 moles of ATP per turn. Okay. We've got hydrogen ions being liberated, we've got CO2 being liberated like we have here. Where the hydrogen ions go, they go to the electron transport chain. Okay. The purpose of the electron transport chain is to generate metabolic water and ATP. The end result of all this, using glycolysis, so that the electron transport chain or oxidative phosphorylation is anything up to about 38 ATP, depending on the substrate that we're breaking down. But this example is 34 ATPs. That's a lot better than the 2 and 3, right, in glycolysis. Okay. That's very much a simplified version, believe me. Okay. There's a little bit more of a detailed version here of the Krebs cycle, the citric acid cycle. So what you've got here, there we are with pyruvate. Okay. Like we said, it's been broken down. So we've got 2 moles of pyruvate being broken down into 2 moles of acetyl-CoA. We've got CO2 being generated. We've got hydrogen ions being generated. They're being picked up by those coenzymes, NAD, NADH. And they're going on to the electron transport chain. Acetyl-CoA enters the Krebs cycle or the citric acid cycle. Okay, the purpose of the citric acid cycle is to generate ATP. Okay, we've got a lot of these intermediate substrates that are broken down under the action of a lot of different enzymes. Okay, for this class, you don't need to know the names of these particular enzymes or even particular substrates. Like the 2 important substrates that I do want you to remember are succinate dehydrogenase. Okay, 1. And I think it's citrate synthase is another important one. Okay. I'll tell you why coming up. So, with one turn of the citric acid cycle, we're getting 3 NADHs, 1 FADH2, and 1 ATP. But remember, you have to multiply that by 2 because that's for 1 mole of acetyl-CoA entering. You've actually got 2 moles of acetyl-CoA entering. So actually, you multiply that by 2, you've got 6 NADHs, 2 FADHs, 2 moles of ATP. Per turn, 2 moles of acetyl-CoA. Okay, this is pretty much just saying what I just said, okay, but we're writing down, so I'm not going to go through that. What we're going to talk about coming up in a second is what the fate of NAD and FAD, or NADH. These carry hydrogen ions to the electron transport chain. So the NAD and FAD, like we say, become NADH and FADH2. So, yes. Okay, so what's the purpose of the electron transport chain? One is to make metabolic water, the other one is to generate ATP. It gets very confusing. It's hard to understand. Okay? So, in essence, electron transport chain splits NADH and FADH, producing hydrogen ions. It liberates hydrogen ions, which are recombined with oxygen to produce water. We'll see that. The electrons produced from the split of NADH and FADH provide the energy for phosphorylation of ADP to ATP. And 1 mole of glycogen can generate up to 37 to 39 molecules of ATP at the end of the electron transport chain. That's oxidative phosphorylation. Okay, this is the diagram of your book for electron transport chain. I think it's terrible. I don't like it. I'm not going to use it. And there's another diagram I am going to use. Okay. What I've got here basically is just, you can go to the web, and I've put the web pages here. There's 2 animations you should watch. And in particular, the one, I believe it is this one here, one in bold. That's a very, very good animation of the electron transport chain. I would highly advise you to go to it.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Okay. I'm sure that's good in your notes. animation has something like this, but it's all moving. Okay, so it's better. But basically, in essence, what we have here, we have an ADH coming from the cytosol, from glycolysis, right? This glycolysis is happening right up down the cytosol, right? This is the mitochondria, this square portion of the mitochondria. Okay? So the NADH is shuttling hydrogens into the mitochondria. We've also got NADH coming from the citric acid or the Krebs cycle, along with FADH. Okay? The NADH and FADH, if you like, get rid of their hydrogens and they're pumped. They get them off to like complex 1 as an example, and they're pumped from the matrix of the mitochondria into the inner membrane, across the inner membrane, into the inner membrane space of the mitochondria. Remember, the mitochondria has got double membranes. Okay? So that's the first thing that occurs. Then we have these other complexes, and also electrons are transferred and are carried down, passed along from these complexes, complex 1, complex 2, through coenzymes all the way down to complex 4.
What are the complexes?
Complexes are basically shuttling proteins. Okay, shuttling proteins. Okay? So you've got, and every time an electron is shuttled from, say, a complex to another complex, hydrogens are pumped into the inner membrane space. And that becomes important because there's going to be more hydrogens, you can see there's more hydrogens in the inner membrane than there is in the matrix. And that sets up a proton gradient. Or hydrogen concentrations higher in the inner membrane than the matrix. Okay. So what we've got here, once we get down, once we get past cytochrome C, another pumping protein, to complex 4, the hydrogens combine with the electrons, with molecular oxygen to form H2O, metabolic water. Okay? And that occurs in the matrix. Complex 5 becomes a very, very important complex for us. The hydrogen ions move from the inner membrane space into the matrix in that direction. This is the last complex, a very important complex. Okay. The animation goes into a little bit more detail than I am, that's fine. Okay. And that hydrogen concentration creates an energy gradient that recombines ADP of inorganic phosphate, located obviously in the matrix, and make ATP. So it's the energy from that hydrogen concentration recombines ADP inorganic phosphate with ATP. And that's the end result, if you like, of the electron transport chain. ATP. Making of. Okay? But absolutely make sure that you go through that animation. So like we said, if you combine glycolysis with the electron transport chain, you get around about 37 to 39 ATPs. Okay? I don't expect you to know this. This is just there for your entertainment value. Okay? Knowing where do all these ATPs come from. You can go through this yourself. It should make sense. There's glycolysis right here. There's the Krebs cycle. Where are all these ATPs coming from? Well, this goes through it for you. Okay?
So we don't need to know exactly where each one is coming from, but we should know how much of everything the Krebs cycle kind of costs.
Exactly. If this was a biochemistry class, you would be expected to know this. Okay, it's not, so I don't expect to know this. But I do expect to know things like roughly how many ATPs are generated within exoxylated phosphorylation. Like I said, I want to understand names of some of those enzymes in the Krebs cycle. Okay. Etc. Okay, so let's leave the electron transport chain, oxidative phosphorylation. for a minute and talk about the cellular uptake of glucose, because that comes important to us. So, the question is, right, let's go back and say, okay, we've got glucose in the blood, right? Here's the blood, here's the muscle cells. How do we get glucose into them? How do we actually get glucose into the muscle? Okay. Well, depends on insulin. Depends on insulin. So we have an insulin receptor. So you know, right? Glucose increases in your blood, insulin increases in your blood. Where's insulin coming from? The pancreas. So glucose is heavily monitored by the pancreas, of course. So you get a big bolus of, you eat a lot of carbohydrates, breaking down the glucose, and insulin is obviously secreted in the pancreas. Must still have a lot of insulin receptors. Bound to these insulin receptors, what we call the glucose transporter proteins. There's others, the muscles glucose. Okay. So when insulin is high, because you've got a little glucose in the blood that's high, the glucose transporter translocates to the cell membrane, muscle cell membrane. Start with that. Once it does that, glucose can enter the muscle cell through the glucose transporter. You know that entering here really looks entering like this. Okay. And then you know obviously the fate of glucose, right? It's either broken down or stored in glycogen. So we've got to have sufficient amount of insulin, glucose transporter transportates in that way, glucose enters the cell. Okay. Let's switch gears a little bit and talk about fat. Okay. Another term you have to become familiar with, lipolysis. The breakdown of triglycerides and glycerol and free fatty acids. Remember I just had that diagram on the board. Completed with the free fatty acids, that's lipolysis, lysis breaking down. Okay. Free fatty acids travel via blood to muscle fibers and are broken down by enzymes in the mitochondria into acetic acid, which is then converted to acetyl, which is then converted to acetyl-CoA through we term that metabolic pathway beta oxidation. The oxidation of fat, beta oxidation. Acetyl-CoA enters the Krebs cycle and the electron transport chain, which we've just been through. Downside is fat oxidation requires more estrogen compared with glucose. Why? A free fatty acid molecule contains more carbon. So you need more oxygen to break it down. Okay? The upside is, the good news is, you generate a lot more ATP when you break down fat than oxidative phosphorylation of carbohydrates. So this particular palmitic acid can generate up to 129 ATPs compared to about 39 ATPs from breakdown of glycogen. Okay. So where, for instance, is a triglyceride molecule broken down? Well, a number of places. VLDL, very low density lipoprotein, triglyceride molecule. It can be broken down actually in a blood vessel. And the actual lipoprotein lipase, the enzyme. And it's broken down like I had up there, that easy schematic, free fatty acids and glycerol. Okay? Free fatty acids can then enter the cell, the muscle cell. They can be recombined with glycerol, okay, to make a triglyceride molecule. And the term for that is lipogenesis, to make it known, right? Okay. They can also combine with CoA, coenzyme A, to make free fatty acid coenzyme A, that can then enter into the mitochondria and be broken down under the Krebs cycle. Electron transport chain under the action of beta oxidation. And also glycerol. If you have high glycerol in the blood, that will trigger epinephrine release of the adrenal medulla. Okay? That's a hormone. Binds to a receptor on the cell membrane. We get the trigger, we get the release, we get the making of cyclic AMP, which we'll see later on. And that will also be responsible for breaking down triglyceride molecule. Okay? So there's many different ways and places that triglyceride molecule can be broken down. And then obviously with breaking down, there's a breakdown of triglyceride molecule, it's broken down as 3 fatty acids, glycerol again, and then protein, don't spend much time on protein, we've already mentioned protein. Spoke about what we use during exercise. Unless you're in a starvation state. So we did say that some amino acids can be converted to glucose or intermediates of oxidative metabolism. The term for that, we'll see that coming up as gluconeogenesis. Schematic out of your book here. Common pathways for the metabolism of fat, carbohydrate, and protein. So what we have here, we have glycogen, which we've already spoken about. Glycogen broken down to pyruvic acid, acetyl-CoA entering the Krebs cycle. Okay. Triglycerides, fatty acids, we've already said, combined with acetyl-CoA, free fatty acid acetyl-CoA, but also enter the Krebs cycle. Glycerol combined with phosphoglycerol aldehyde and enter glycolysis right here. Proteins can be broken down into amino acids and also can be combined with acetyl-CoA and enter the Krebs cycle. Okay. You may have heard the term before, you may not. Fat burns in a carbohydrate flame. Fat burns, what do you mean by that? Fat burns in a carbohydrate flame. This is that. So to burn fat efficiently, you've got to have sufficient carbohydrates. That would make sense. In other words, you've got to have sufficient acetyl-CoA to combine with free fatty acids and enter the Krebs cycle. If you have low amounts of acetyl-CoA, you can have low amounts of free fatty acids acetyl-CoA entering the Krebs cycle. And the same up here as well. That's what we mean by fat burns in a carbohydrate flame. Okay. We were speaking about that. Okay, oxidative capacity of muscle. Oxidative capacity of muscle, QO2. It's kind of akin to VO2. Maximal oxygen consumption, whole body oxygen consumption, we're talking down at the muscle. We can measure this in the biochemistry lab. It's very, 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 very difficult to do. You can measure it. The oxidative capacity of the muscle. How much that muscle is consuming oxygen. How much oxygen is being consumed. Okay? Some of the enzymes that we can measure, or representative enzymes to measure this oxidative capacity. Remember I mentioned those before, succinate dehydrogenase, citrate synthase, those are located in the Krebs cycle. We can measure those. Okay? What determines oxidative capacity? Well, obviously, oxidative enzyme capacity within the muscle, if that goes up, oxidative capacity goes up. Biotype, composition, and number of mitochondria. Okay? Increase number of slow twitch, increase the number of mitochondria, which is an adaptation to endurance training, and then increase oxidative capacity of the muscle. Well, there you go, endurance training, right? And obviously oxygen availability and the uptake in the lungs. That's going to become important. When you go up to altitude, your oxidative capacity goes down. We're going to talk about that. We're talk about altitude physiology. So is there a relationship between then oxidative capacity, this is real data here, oxidative capacity in the muscle, and muscle succinate dehydrogenase enzyme activity? Yes, there is. It's a very tight relationship. You increase that enzyme, increase muscle oxidative capacity. Okay. It's not out of your book, but it's a very important diagram here. So, some of the factors then that influence the rate of oxidative phosphorylation or oxidative capacity, it's the same thing. Okay. What can determine the rate of oxidative phosphorylation? So that's glycolysis, electron transport chain. Okay. Obviously, the activity of oxidative enzymes in the mitochondria. Okay. By that we mean the concentration. You increase the concentration, you can increase activity, you can increase the rate. Enzymes, very important. Oxygen availability in the mitochondria is going to play a role. More oxygen we can get down there, can increase the rate of oxidative phosphorylation. What determines that? We're talking about this again in cardiovascular physiology, the transport of O2 to active muscle, of course. And that's dependent upon the rate of blood flow to the active muscle, cardiac output, etc., etc. So you can follow this flow chart all the way back. It should make sense to you. Okay? The two big ones, of course, then are activity of the oxidative enzymes and the transport of O2 to the active muscle are big players in determining the rate of oxidative phosphorylation.